Thank you very much, Professor Legato, for inviting me. I thanks also the MRA Foundation. And I also thanks the speakers, because thanks to their previous talk yesterday and this morning, this will help me to skip some slides that to avoid the additional information that are not new, so I can go towards the topics of my lecture. We know that female and males show large differences in susceptibility to complex disease. We know in the past cardiovascular disease, and in particular myocardial infarction. In the past, we consider myocardial infarction as a prerogative of the male sex. Unfortunately, during the last decades, myocardial infarction in male and female reach the same levels. But unfortunately, again, female experience the worst prognosis, and particularly for the negative effects of the acute events linked to the remodeling of the wall. This may be due to several reasons. We know that all trials have been performed just on male sex. So we now can consider female sex as orphan of dedicated therapy to contrast the remodeling of the wall of the heart after myocardial infarction. Other complex diseases, such as autoimmune disease, yesterday one of these colleagues spoke about the lupus erythematosus systemicus, and this is a prerogative of the female sex. But not only autoimmune disease in the strictly sense of the word, also multiple sclerosis that has a strong autoimmune basis, but share also a inherited predisposition. For example, for the iron genes, because iron overload, local iron overload, for example, in the brain, may be responsible for increased oxidative stress in the brain. And we know also that female may have, with respect to male, different iron levels, as well as different folate level, as well as different B12 and B6 level, that together are responsible for the worst prognosis, in this case, of multiple sclerosis patient in the female. Last but not least, neurodevelopmental neurological disorder. We have just stated that the male-female ratio in autism spectrum disorder is 4:1 towards the male sex in this case. But a more realistic ratio, sex ratio, is less, 2.5:1. This is a speculation, but we have reason to think this. Because if we, as for myocardial infarction, receive an imprinting in our mind of the community that myocardial infarction is connected to a problem to the heart, the chest, because these are obtained by a male casuistic. The females when have different symptoms do not think that is it an acute myocardial infarction. So this start a series of consequences that she will pay in the prognosis, in the future prognosis. The same maybe for autism. According to the theory of the developmental origin of health and disease, susceptibility to this complex disease arises early during our life. And this period, of course, is in the home life, in the utero life, during the pregnancy. The two mechanisms that may link the environment stimuli, the internal stimuli, the genetic stimuli due to the genome differences, are the epigenetic mechanism. Genomic imprinting and X chromosome inactivation are the most to investigated. I will skip to explain you the mechanism because now we know. But it's important that those genes that are protected from the wave of global demethylation approaches that occurs immediately after the fertilization, before the blastocyst exist are influenced by external factor but depend from two completely different mechanisms in the male hemigenome and the female hemigenome. We will see in the next slides additional information on this. This picture show that almost all chromosomes contain genes that may be protected by the demethylation program that happened at the beginning. The blue peaks are the chromosomes that contain genes from the paternal hemigenome, and the red one are the genes from the female. The gray one are those that do not contain a appreciable amount of gene that can escape this. We know also that the loss of imprinting of the differentially methylated regions are responsible for the classical neurological disease like Prader-Willi, Angelman, Wiedemann disease, Russell syndrome, and so on. So this is an important and crucial point to discuss also for autism. What does it mean that in the X chromosome may exist genes that escape the inactivation? The skewed inactivation is not something that is on-off. We can have some genes that are more prone to escape, other ones that no. And we know that as baseline, we can consider that an appreciable amount of genes, over 15% of X-linked gene, escape the inactivation. So this means that in the same cell, or female, of course, the two alleles cooperate together to the expression, also in the inactivated chromosome. And again, more than about 20% of X-linked genes are associated to neurodevelopmental disorder. I will not explain you how access works because we discussed it this morning. But the only thing to add is this. On your left, you see that female, in each tissue, in each organ, in each cells, may be considered a mosaic of the two different expressed X chromosomes, male and female. This potentially may be a protective factor because 2 is better than 1. So just in case of mutation that reduce or abolish the expression, they can have in this non-random or random inactivation 1 chance more than male that have just 1. Conversely, methylation can appear throughout the genome and may be present and occur also in different gene structure. It's easy to understand that a methylation in the promoter can finely tune the expression level of the G product. Moreover, methylation profile change during the life of a people. Normally, physiologically change, starting from the in utero life. We know also that within the same people, the same individual, to be health or disease patients is a condition that carry on the change in the methylation profile of the same individual in the same moment. And finally, methylation change, of course, also between the two sexes. We investigated in our laboratory a group of neurodegenerative diseases. In this case, Alzheimer's disease patients and mild cognitive impairment disease. We split the group of patients just by sex. This was the only difference. The disease was the same. The disease onset was, the mean disease onset of the group was not different. And also the minimal mental score disease, the clinical tool to ascribe physical and or mental disabilities. But when we look at, when you assessed the methylation profile, we found that female group had a significantly lower global baseline methylation. But what changed? In a follow-up, the female group had a rapid progression of the disease. So, at the end, they developed a more severe disease, starting from the same disability score. The epigenetic clock that start to work at birth, but we can think that any cell start at the beginning of the S formation. So also the different gametes, male and female, may have different start clock. But we think that after fertilization, the timers stop down and we start again. We don't know. We must compare during our life that we cannot consider hypermethylation wrong or good and hypermethylation wrong, a good thing or a bad thing. We should consider the global assessment, and we can have different part of a gene, different part of the genome, hypo or hypermethylated according to the condition of the patient. So we must compare the methylation profiles in a vertical line, I mean younger, older, and between left and right, and again between the two sexes. Thanks to this information, we can move with different rates. We can have a slowed aging on your right and an accelerated aging. Thanks to what? First of all, inherited predisposition that can help us or not, but thanks also for the environment situation. That is the thing that we experience during our life, the concomitant disease, lifestyle, and so on. Also wrong style habit, low activity, physical activity, low mental activity. These are risk factors, of course, for physical and also for the mind. All these processes that we are discussing are essential for the correct fetus and individual development. And among the several tissue cells and organs that are affected by the methylation, by the genomic imprinting, the epigenomic imprinting, the brain is the most sensitive, and is the neural cell that is the cell that, starting from the beginning of the organization of the development of a brain, may be affected by a wrong homeostasis. Many neurodevelopmental disorders may originate from a defective signaling during fetal and perinatal brain development. We know, this morning we spoke about this, placenta has a sex. And this influences the fetus, that of course share the same sex of the placenta, not only in the development during the 9 months of the pregnancy, but also in the future life, when after birth, and not only on himself, but also influence the health condition of the mother during the pregnancy and after the pregnancy. The degree to which genes are subjected to X chromosome inactivation can influence the neurodevelopmental disorder phenotype between the two sexes. This table I get by the paper from Brand et al. Of 2021. In the original table, there were a lot of genes. I selected this just because all are on the X chromosome, all undergo to the X chromosome inactivation, quite all. But when we compare the clinical phenotype between the two sexes, we can observe different. Just to comment with you one. The first one, the NLG entry. This is the example of two opposite clinical phenotypes. The same gene, the same chromosome, the same disease, but male are considered affected patients. Female are considered unaffected carriers. This is not the only one gene that is responsible for this behavior. You can see also the UPF3B. But another thing is that when the disease is present, the clinical signs are different. So a complex disease also for this. But what kind of unit is responsible for the methylation action on the genome? And another important question is this unit differently handled by the different sex? Yes, not only for intrinsic reasons, I mean genetics, but also for the different amount of cofactors that are different also. When you assess laboratoristic analysis, you have male and female with different normal range, the so-called normal ranges. The placental sex seems one of the major determinants in response to perturbations during the pregnancy. And not only for the fetus, but also for the mother, and not only during the 9 months, but also after, for himself and for the mother. Again, male but no female placentas, so XY, seems responsive, more responsive to external change and internal change, or we can call perturbation in the maternal environment. I mean the micro environment in the in utero microenvironment. I mean diet, folate, stress, comorbidity, infection, and so on. So the placental sex as a role. Normally in laboratory, we consider that the methylation pattern of the different tissues and organs are the same. This is a good thing because when we usually investigate the whole blood, the peripheral whole blood, we hypothesize that the brain have the same global methylation. In general, we can accept this, but for the placenta, we know that the main crucial genes undergo to opposite methylation. And this may be important according to the sex that is within the placenta. Placenta-specific imprinting and fetal sex can modify the course and complications of pregnancy, may have impact on maternal health and well-being both during and after the pregnancy. So we have to speak about the conflict hypothesis. I'm not completely I don't agree completely with this hypothesis, but is a good message just to understand some basic information. If we think that the two different genomes, male and female, speaks to the fetus genome, and to the fetus itself, we can hypothesize that the male genome suggests to his child, you have to grow big and strong, and you have to use all the maternal resources you can. Because, not only for this I hope, future babies may not have my alleles. This may seem an egoistic way to think, but the selection of the nature probably find this a good decision. On the other hand, when we ask to the genome of the mother the same questions, she replied as follows. Dear baby, you should grow well, perfect, but please leave me some resources for myself and for my future babies that may be from the same father or not. The two statements of the female genome are not egoistic. Why? First one, because when the female genome say to the child, please leave me some resources for myself, she is thinking to the baby. Because the mother, after the birth of a child, have to preserve energy and a well health status to help the child to grow up. So it's something that, yes, is important for herself, but is more important for the child itself. And also the other message is for my future babies, otherwise we cannot have the evolution and we cannot be here now. So these two opposite conditions, together with the fetal sex in the middle, are responsible for the outcome of the pregnancy. Altogether, this message can say us that all the difference that we observe during pregnancy according to the fetal sex or according to the placental sex can be related to this way to work together. The unit that is responsible for this crosstalk, for this speaking between maternal, paternal, and the fetus genome, according to the topic of my lecture, are the one-carbon metabolism units that came from the folate. But as we know, other mechanisms are involved. But just to be rapid and give you a message, the amount of one-carbon unit bridged, linked to the different region of the same gene or to different region throughout the genome are also responsible for the different three-dimensional architecture of the genome. And we can access to open or closed chromatin association that are responsible respectively for facilitation of the synthesis or not of the gene product. All the mechanisms that I've discussed till now happens during the complete life, our life, starting from the fertilization step, that is the first one. But again, also during the first steps, we can see completely different mechanisms of what? Of the raising of all the messages and information that the two epigenome, male and female, have according to the diploid genome of their parents, that are the grandparents of the fetus. So we have to erase this suddenly, immediately after the fertilization, the zygote, and this concludes with the blastocyst establishment. And the second outstanding action is that now the genome of the mother, the in utero microenvironment, is responsible for a new project, a new design, a new painting. Is the mother that now, starting from zero, paint the new reprogramming, the new remethylation messages to this white table. And even if we think that the same blastocyst and the same mother the blastocyst is implanted in the utero with the same mother in different period of her life, this picture, this paint is completely different. So it's not only important the genome, of course, but if we have several clones of a blastocyst and we introduce in the utero of the same mother in different time of her life, the new painting will be different. And again, during the 9 months of the pregnancy, the painting continues. The mother, according to internal, external stimuli, stress, mental, physical stress, or different stress factor, good situation, not only negative situation, all these cooperate to define the metyloma of the child. And again, among the more cellular tissue and organ, the brain is the one that is more sensitive to these several actions. And again, after birth, during the infancy, puberty, and adult life, additional external and internal stimuli contribute to the plasticity of the metyloma in the brain and also in the other organs of the child, boy, adult individual. And now I can speak about the one carbon unit in quite detail. This is the folate cycle. Don't worry for the several enzymes that are synthesized by different genes, and these different genes are extremely polymorphic in the population. Please look just to these two ones, DHFR and MTHFR. Probably you know because several papers on autism investigate probably the MTHFR more than the DHFR. Anyway, these two enzymes are reductase enzyme and are responsible for the partial activation or the full activation of the 1-carbon unit in the folate. Folate is not just one molecule. Folate is characterized by a lot of isoforms. The most active is this one, the 5-methyl tetrahydrofolate. But it's the last molecule within the cell that is helpful for the methylation program. But the isoform, so-called partially activated or intermediate products, are fundamentally important for the novel DNA synthesis. So, when we speak about cancer, in which a faithful and optimal neo-DNA synthesis is important to avoid mistake, we need the pre-activated isoform of folate. These are the precursors of the nucleotide, AGCT. And when we have low amount, intracellular low amount of this precursor, we can have other chance to experience any kind of cancer. But we cannot decide, the cell is a hard task for the cell to decide it's better to have a more faithful DNA synthesis or it's better to have an optimal methylation. Also because before reach the final target, the epigenetic modifications, I mean DNA methylation, there is another important task for the enzyme of the folate cycle. That is to lower the homocysteine within the cell. We know that homocysteine is a toxic, particularly neurotoxic amino acid because it has two sulfur residues. So, thanks to the active isoform or folate, homocysteine can go down, so we skip the risk hyperhomocysteinemia condition, and merely by the adding of the 1-methyl group to homocysteine, it became methionine, a very interesting amino acid. And this is one of the last donors of the 1-carbon unit to the DNA methyltransferase enzymes. And this happened thanks to SAM and SARC, acetylcysteine homocysteine and acetylcysteine methionine. And this is very important for the talk that we had just now to investigate the role of folic acid supplementation, the role of the active isoform, I mean the 5-methyl tetrahydrofolate, or why not the SAM. Additional vitamin of the B group, such as B6, B12, and B9, the folate itself, cooperate to this because the most interesting enzyme for methionine synthesis is the MS. This one. The methionine synthesis is the enzyme that receives from the MTHFR, the methyl group, they donate to the methyl group, link to the homocysteine and transform it in methionine. I told you at the beginning of this slide that these genes are extremely polymorphic. And these are functional polymorphisms, not intronic, also intronic but functional at the same. On your left, you can see that the most common polymorphism also often investigated in autism cohort of patients, the MTHFR. You can see that the specific activity of this enzyme decreased significantly as the number of the polymorphic allele increased in the genotype of patients. The homozygous condition for this polymorphism, I mean TT, on your left, the red block. The TT homozygous condition is present in no less than 10% of normal general population, reaching also increased amount and rate in autistic patients. So, contrast results, but a lot of paper conclude that the homozygous condition of this enzyme are overrepresented among autistic patients. This means that the active folic acid folate is important for DNA methylation of the brain. But when we have a low activity of this enzyme, we can obtain a more faithful DNA synthesis. So the question is better an optimal DNA methylation or is better the optimal novel DNA synthesis? We can change enzyme. The DHFR is the first one that received from the diet, the folate, and after the receptor, decide to apply two reactions of reduction to obtain those kind of isoforms that are responsible for the faithful DNA synthesis. Another common polymorphism with the same rate among the general population has an opposite behavior. The expression rate of this enzyme increased as the number of the polymorphic allele increase in the genotype of patients. Being two common polymorphisms, these two conditions may coexist in the same individual. And so we can assist to complete this unbalanced situation. For example, very low activity of the MTHFR combined together with the overexpression of DHFR is responsible for a lot of isoform for the DNA synthesis, no isoform or very, very low isoform for the DNA methylation, and also the opposite condition of the high activity of MTHFR that move towards the methylation and we remain in the cell without isoform for an optimal methylation. So the two conditions are risk factors for, respectively, neurological disorder or for, in general, for cancer. Now, the last two slides. I cannot see the time. Oh, sorry. The last two slides. We investigated in a group of patients, children affected by pediatric leukemia, the presence of these two polymorphisms. And the interesting thing that we found is that we know that the peak or onset of pediatric leukemia is 2-3 years, very early. We found that according to the fact that the protective allele, the protective haplotype, I mean DD, give us two different and opposite situations according to the fact that this protective egoistic gene was carried from the mother or from the child. When the child carried the protective, the mean onset age of the group of this carrier is very high, about 8 years. So the mean level of the age shift. But when the same protective genotype is carried by the mother, you can observe a drastic and significant early onset. And this trend, 8, 6, 5, is opposite to the red one, 5, 6, 8. Finally, the missing haplotypes. We know that according to a classical natural pregnancy, we cannot have these two opposite situations in the fetus and in the mother. We cannot have a fetus that is DD homozygous and a mother that is homozygous for the absence. The maximum situation is the heterozygous condition. And since we, in the heterozygous condition, are able to find the three different mean onset for the pediatric leukemia, and we can see that according to the number of the allele, the mean age onset of the patients is shift to left, to right, to increase. The missing haplotype is that we cannot obtain this genotype in the fetus when the mother has the opposite situation. But, yes. But we can find the missing haplotypes thanks to heterologous ordination. When we obtain a pregnant woman, according to the fact that the oocyte came from an external heterologous donation, the oocyte can carry one allele, because they are haploid, that when met in a heterologous mother, the other allele can have this rare situation. I mean, the fetus and the mother may be in conflict or may cooperate together. We don't know, because we can solve this problem or by analyzing retrospectively those people born thanks to the overdonation, or in animal models with rat. And we can also decide and try to investigate if in natural condition, one of the two haplotypic conditions may be a risk or extremely protective. And the last information that could be obtained by this investigation is that we can finally ascribe to what kind of genotype may be helped for folic acid supplement. Because some combination of different haplotypes in the mother and fetus should not receive folic acid supplement, because may be a risky factor. I conclude with this. That is a sex-specific maternal placental fetal interaction, including sex-specific transplacental crosstalk with potential significant biological implication, even longer times, I mean after birth, certainly with genetic and epigenetic involved mechanism. Thank you very much.